Hello and welcome to JCO Precision Oncology Conversations, where we bring you engaging conversations with authors of clinically relevant and highly significant JCO PO articles. I'm your host, Dr. Rafeh Naqash, podcast editor for JCO Precision Oncology and associate professor at the OU Health Stephenson Cancer Center at the University of Oklahoma. Today, I'm excited to be joined by my friend, Dr. Tejas Patil, assistant professor of thoracic oncology and associate director of clinical affairs at the University of Colorado Cancer Center, and also the lead author of the JCO PO article entitled CRESTONE, Phase 2 Study the Efficacy and Safety of the HER3 Monoclonal Antibody, Seribantumab, in Solid Tumors with NRG1 fusions. At the time of this recording, our guest disclosures will be linked in the transcript. Tejas, welcome to our podcast, and thank you for joining us today.
Rafeh, thank you so much for having me. It's a real honor to be part of this and to honestly just to catch up and chat with you, do some science, and yeah, let's take it away.
Absolutely. I know we met a couple of months back, and I was really looking forward to this specific podcast. Oftentimes in JCO PO conversations, we'd like to discuss rare entities. And today's podcast is very much about a rare entity called NRG1 fusion. So given the breadth and diverse audience that we have with trainees and community oncologists, can you tell us about what NRG fusions are? Where are they identified? What is their significance? And I always like to understand a fusion from the standpoint of breakpoints and what that fusion leads to in terms of a protein, functional oncogenic protein. Could you help elaborate on some of those concepts?
Yes, certainly. That's a big question, and so I'll do my best to give a focused response. NRG1 stands for Neuregulin-1. It's an extracellular ligand of the ERB3 receptor. And when we think about NRG1 fusions, the canonical fusion partner that's been described, and that was something we saw in this trial, is CD74. But it's important to note that's not the only fusion partner. There's a huge variety of fusion partners. And basically what you see is, in the case of CD74 NRG1, let's just use that as an example, you have an interchromosomal break that leads to ectopic expression of NRG1 gene downstream of the CD74 promoter. So what you get is this gene fusion that consists of a N-terminal bound CD74 portion and a C-terminal EGF ligand domain. And what's interesting about NRG1 fusions, and this is in stark contrast to ALK and ROS1 fusions, for example, is that the outer cell membrane is what is acting like functionally as a ligand to ERB2 and ERB3 heterodimerization. So in many ways, the gene fusion is encoding a ligand that then promotes oncogenesis. And that's in stark contrast to an ALK fusion or ROS1 fusion, where the gene fusion is actually producing the oncoprotein that's driving oncogenesis. One thing that we have seen is that these tumors tend to be disproportionately overrepresented in invasive mucinous adenocarcinomas. And we see them in pancreas and lung, but they're not only in these. We can also find NRG1 fusions in cholangiocarcinomas. And targeting these has been very challenging. A variety of different pan-ERB therapies have been tried in the past. These include things like afatinib or even drugs like trastuzumab, really with mixed results. So it's a very rare fusion. It's got a very unique biology. One thing I will note is that the retained EGF ligand, we don't really fully know whether it binds ERB3 in an autocrine or a paracrine fashion. I think that's sort of still being sorted out. Nonetheless, it is still functionally a ligand, whether it's autocrine or paracrine. And I think this poses unique therapy design when it comes to developing drugs that can target this fusion.
Thank you for a very detailed explanation. And I know there's been a decent interest in NRG fusions with recent accelerated FDA approval. Could you tell us a little bit more about that mechanism of action and what that data looks like?
Right. So there have been some recent FDA approvals, specifically with a drug called zenocutuzumab. And in many ways, zenocutuzumab and this other drug, seribantumab, the main targeting strategy is to create a monoclonal antibody that blocks the extracellular domain of ERB3 or HER3. And by doing this, you effectively block the NRG1 fusion ligand from binding to ERB3 and inducing heterodimerization of ERB3, and typically it's ERB3 and ERB2. So the differences between seribantumab is that that's a pure HER3 monoclonal antibody. Zenocutuzumab, which has ultimately led to an FDA approval for this drug, blocks both HER2 and HER3.
And as I try to understand it, sounds to me more like in one mechanism of action, you're blocking the engine, which is the zenocutuzumab. In the other mechanism of action, you're sort of preventing the gas from being delivered that drives this heterodimerization of HER2 and HER3. Is that a fair way to understand this if you had to simplify this whole concept?
I think they're more similar than dissimilar. So I do think that fundamentally what's happening with zenocutuzumab, and actually this is the similar mechanism of action with seribantumab, is that they're blocking HER3 and from the extracellular side, thereby preventing NRG1 like ligand from binding to the extracellular domain. And so the analogy I think that's probably more apt is the distinction between a drug like afatinib, which is a pan-ERB inhibitor, but with a preference and has been studied in EGFR, which blocks the intracellular tyrosine kinase domain, versus a drug like cetuximab, which is a monoclonal antibody that blocks the extracellular domain, and in theory blocks EGF-like ligands from binding. So I think zenocutuzumab is sort of more in the cetuximab camp, if that helps from understanding it from a mechanistic standpoint.
Sure. And then looking at your specific trial, could you help us understand what the primary objectives of this study were, why this trial was conducted? Was it to improve upon results that we've seen with zenocutuzumab? Was that the idea? And what was some of the patient population that you chose for this study?
Yeah, so this was a multi-center phase 2 clinical trial looking at a drug called seribantumab. This is an anti-HER3 monoclonal antibody, and it was dosed at 3,000 milligrams IV weekly. Because NRG1 fusions are seen across a variety of solid tumors, this was actually a pan-solid tumor study. And in the results, we'll note that there are actually many patients from different solid tumors that harbored NRG1 fusions. The primary endpoint of the study was objective response rate using the RECIST 1.1 criteria. And then there was a variety of secondary endpoints, which included safety, duration of response, progression-free survival, overall survival. When we were thinking about this study, I think one of the things to note was that about 54 patients were enrolled, which is a lot. These are extremely rare fusions with an incidence of about 0.5 to 1%, with an enrichment for invasive mucinous adenocarcinomas, which tend to be seen more in pancreas cancers and in non-small cell lung cancers. So a total of 54 patients were enrolled. There were 9 different tumor types. There were 19 different NRG1 fusion partners. So this gets to what I was talking about earlier, which is that even though we see CD74, actually there's a huge range. So you can't just rely on that. And for the 29 patients that were included, I can certainly talk about the results, but part of the reason we did this study was in many ways, this was born out of the experience with, or at least the hope of this design was similar to what we saw with NTRK fusions, which is that if you find a genomically actionable fusion and you develop a highly effective therapy, you can generate deep responses. Now, I think we learned a lot from doing this study that there's probably challenges with targeting NRG1 fusions that are not as one-to-one to NTRK fusions. And I'm happy to talk about that some more if we want to, or we can go wherever we want.
Sure. Let's take a deeper dive into some of those aspects.
I think the differences between an NTRK fusion and an NRG1 fusion actually come down to the biology. The drugs that target NTRK fusions are intracellular kinase domain inhibitors, and so they're TKIs fundamentally. And when you looked at some of the original studies for NTRK fusion, then part of the reason it got a pan solid tumor approval is if you look at the waterfall plot for those studies, I mean, you're getting objective responses that are greater than 70% across multiple tumor types. Now, that was the hope when we designed studies like this, and certainly that was the hope when I was participating in the seribantumab clinical trial. I think one of the things that's tricky about blocking the extracellular domain and some of the mechanisms of action is that to what extent are you getting ERB3 and ERB2 heterodimerization, and how are you effectively blocking that? That's essentially the strategy that zenocutuzumab looked at. There's other ERB family members that can dimerize. So this is not a pan-ERB inhibitor. This is really just focused on anti-HER3. There were some challenges. I think the other effect that we started seeing when we were doing this trial was essentially what was like a rebound effect, which is that you need to persistently block the anti-ERB3 extracellular domain with drug. And the minute you let off and give patients a treatment break, you start getting signaling rebound. And so it made dosing and picking an optimal interval strategy, whether we do weekly or Q2 weekly, very, very challenging.
I see. From an efficacy standpoint, did you see what you were expecting you would see, or was it a little less exciting?
I think the honest answer is I didn't know what to expect because this is such a rare fusion and we really had no precedent for this type of approach. So I think we were coming into this very open-minded. When I look at the data, the objective response rate was 35%. 2 patients had complete responses. And I think the time to onset was relatively quick. It was about 2.9 months with a median duration of response of about 8.1 months. The thing that I thought was most interesting was that the patients that had the complete responses were patients with non-small cell lung cancer, and the patients that had either partial response or stable disease were those that had pancreatic cancer. But we did see patients with biliary tract cancers that had a partial response. So I think one thing to note is that if you find an NRG1 fusion, especially in the GI malignancies like cholangiocarcinoma and pancreatic cancer, I mean, these are susceptible to this type of therapy. But again, these are small numbers. And I think we have to be very cautious in extrapolating benefit.
So one of my questions that I was going to ask you, Tejas, is from a mechanistic standpoint, downstream inhibition of mTOR, PI3K pathway, would it make sense to combine one of these upstream inhibitors with the combination strategy with downstream inhibitor to achieve better disease or tumor control? And another part B of that question is, did you have paired biopsies? If you did have paired biopsies, the potential to look at phosphorylation markers for like phospho AKT. There's a few others such as EBP1. If you see or would see some downstream inhibition suggesting that it actually blocks that pathway and there's no bypassing of that pathway after a certain duration of inhibition?
That's a wonderful question, and one that I would love to see supported in the context of a trial, because I do think even with the zenocutuzumab data, the drug that is ultimately been FDA approved, what would be helpful to know based on your suggestion is, what if you block the extracellular domain and also block some convergent downstream pathway from the ERB receptor? And the AKT-mTOR pathway seems like a very logical step. So I would be excited to see a study like that. I think one of the challenges is going to be that this is an insanely rare population. I mean, it took a long time to enroll 54 patients across all solid tumors with multiple PIs at multiple sites. So I just think this will be a very hard study to ever do, but I think conceptually I like it and something I would very much want to see more studies do that are in this space. In terms of samples from the study, we had a couple. I know one of my patients had a post-treatment biopsy. We're sort of sitting on them. One of the investigators at MSK, Alex Drillin, and I had some conversations about what to do with these. So, I mean, I think there's some room and potential opportunities here to look at whether some of the downstream phosphorylation changes that you've hypothesized might be in place actually are there. I think that's an interesting scientific question. I think the fundamentally it always comes down to funding and who's gonna pay for this. But yes, I think this would be really something I'd like to do.
Maybe that could be your subsequent publication in JCO PO, and we can have a podcast about it again.
Yeah.
But going back to the AEs, could you tell us a little bit about the adverse events? It didn't seem like it was a laundry list of AEs, especially AEs that would affect quality of life, not just the grade 3 AEs. Could you tell us briefly about that?
Yeah, absolutely. So the most common treatment emergent adverse event was diarrhea, which was seen in about 52% of patients. But this is important. The number of grade 3 events was 6%, and The number of treatment emergent adverse events that led to treatment discontinuation was 7%, and any adverse event that led to dose reduction interruption was 35%. So I think that probably some of this might be related to off-target ERB1 adverse events. There's also a 32% incidence of rash. It's noted in the study, although I'd have to say in the patients that I treated on trial, this really wasn't as common. I think more common was what I saw was hypomagnesemia. That was kind of an interesting signal, and we saw a little bit of with this drug. But nonetheless, my experience with this, and this also tracks with what we see with the zenocutuzumab data, is that it's a pretty well-tolerated drug. And, especially for patients who are having something like metastatic pancreas cancer, in comparison to the FOLFIRINOX that they may have received before, this is certainly a much gentler second-line treatment, certainly more gentle than gemcitabine and Abraxane.
Sure. Based on the lessons learned enrolling on a trial like this with rare fusion, what are some of the maybe 3 next steps that you would take forward in terms of trial design, in terms of rare fusions, rare alterations in general, or any other aspects that you would maybe do differently?
Great question. So I think the first observation from this study is that diagnosing NRG1 fusions is the most important step. And here it's critical to use high quality genomic assays that have both DNA and RNA-based NGS. The RNA in particular is really important because, as I mentioned at the beginning of this podcast, you don't always know what the fusion partner for an NRG1 fusion is going to be. CD74 is the most common, but it is not quite the same as ROS1, where it sort of overwhelmingly represents the fusion partner there. NRG1 has a huge range of fusion partners. And so really focusing on the RNA product in your genomic testing is going to be absolutely critical to finding these. That's step one in my view. Step two from a trial design is that these were really challenging fusions to identify. That being said, I think both studies, the CRESTONE study and the NRG study that got zenocutuzumab approved, I think did a really good job of working closely with our diagnostic partners, either in companies like Tempus, Caris, so that if an NRG1 fusion was found, there was very clear funneling to these studies and very clear language that, hey, there's this trial going on that's looking at this. So I think that kind of language and alerting the provider or the patient who's getting those reports that there are these kind of studies that are ongoing is crucial. And then the third is really thinking about creative ways, and this is more for us in the academic community, to build genomic registries for these really rare fusions. And here I'm sort of inspired by the registry that's taking place at Dana-Farber for nut carcinomas. They have essentially any patient with a nut fusion, they have a very readily accessible protocol, and you can put those patients and kind of register what you did. And I think we need something like that for NRG1 fusions. They're just so rare. It's going to be very hard to do a prospective study in this population. And I think the more we can share our own experiences with what works, what doesn't work, some of these really great pre-post biopsy observations you had. I mean, I would love to do that in the context of a real world study. That would be huge.
Completely agree with those points. I know there's, I think, another RET registry that Mihail Dea from Farber now, Gustavo Russi has also built. It's also resulted in a lot of real-world analyses. I think we need a lot more for rare fusions, rare tumors aspects otherwise. So thank you again for sharing all those insights. Next minute and a half, I'd quickly like to ask you your career trajectory. From what I know of you, I know you're very interested in precision medicine. So could you tell us a little bit about how you got into that field and what motivates you there? And just talk about the last couple of years, your training and fellowship and academic faculty career, and where you're heading and or hopefully planning to head in the next couple of years.
Yeah, so I did my medical school at the University of Southern California, and then I did all of my training, internship, residency, and hematology oncology fellowship at the University of Colorado Cancer Center. When I was an intern, I worked very closely with my mentor and friend, Ross Camage, and my other mentor at the time was Bob Doble. I think these two faculty were instrumental in building my career and getting me interested in precision oncology. They are both leaders in that space. My first job out of hematology oncology fellowship, actually, I was fortunate to have been offered a position at the University of Colorado. And since I've started here, I've really been interested in working with our translational scientists to think about the management of acquired resistance, thinking about novel biomarker identification. So things like NRG1 fusions, which at the time when I started, I mean, it was a known fusion, but it was no real therapy for it outside of this trial. And then finally, to think about rational therapeutic combinations that are driven by our understanding of evolutionary biology, molecular biology, and to start, as a plea for the entire academic community, to start having novel trial designs, thinking about who do we need to do intensification therapies for based on the biology. So that's kind of what I'm passionate about. That's what I've been working to get grant funding on. I have a particular interest in studying drug-tolerant persister cells after the initiation of targeted therapy. For the audience, this is a subset of cancer cells that survive after the initiation of highly effective tyrosine kinase inhibitor therapy. And what's important about these cells is that they're not resistant in a kind of traditional sense. They're really dormant. They have developed phenotypic adaptations that allow them to survive in the context of a tyrosine kinase inhibitor. And how they do that is not really well understood. And to the extent that we can understand how they do that, I think there are therapeutic vulnerabilities that we in the community can potentially use to our advantage and help patients who might be in this situation live longer lives. So that's where I want to go in a nutshell. I hope that others in the field also feel inspired by doing similar things.
That definitely is very inspiring. Hopefully, you will continue to do most of that work and then publish in JCO PO and other ASCO journals. And hopefully we'll get to talk again. Thank you so much for your time today.
Thanks, Rafeh.
Thank you for listening to JCO PO Conversations. Don't forget to give us a rating or review and be sure to subscribe so you never miss an episode. You can find all ASCO shows at asco.org/podcast.
The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. Guest statements on the podcast do not express the opinions of ASCO. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.